Okay, so Bio 111 students, this is mini lecture 2 for chapter 14, and we're going to focus in this little lecture about how in the beginning of the 1900s, a scientist, a mathematician actually named Punnett, created a graphing or picture device that is really useful for us to very quickly uh, record the gametes that each parent can provide during meiosis, and then the potential genotypes and phenotypes of offspring in a Mendelian cross. So I'd like to go through a Mendelian cross with you, and I'm going to stick with the flower color example because that's the example we used in our first mini lecture when we were going through Mendel's exact experiments. So I'm going to start with basically how you draw a Punnett square, how you construct a Punnett square, and we're going to fill it in based on Mendel's experiment. And then I have assigned you some problems to do that you can find on Canvas in this module, and a step-by-step process for you to convert a word problem into the correct Punnett square. But for today, let's just focus on, for this lecture, let's just focus on how to construct the Punnett square. So, first thing that we're going to do is you're going to draw a square. And inside that square, you're going to place 2 lines to make 4 squares total. Now, in addition to that, you're going to put a diagonal line here, and you're going to extend each of these lines from the top and from the side. On the top and the side, this is where you are going to put the symbols for the gametes. Okay? And I'm going to draw those like as circles to remind you eggs and sperm. And then up across the top here, this would be the gametes from the other parent. Sometimes in these pictures, you will see someone designate with a male symbol that these are the male gametes, and with a female symbol that these are the female gametes going down the side. This is not important, but sometimes out of habit, I just do it when I actually make a Punnett square. Now, it's common practice in Mendelian genetics problems to assign a symbol, which is actually for us going to be a letter of the alphabet, to each of the alleles that are present in an organism. Okay? So I want you to think back to our purple and white flowers from our first mini lecture and remember which of those alleles was the dominant allele, which was the recessive allele. So remember, we took purple flowers, crossed them with white flowers. They were true breeders, and the F1 generation was all purple. That phenotype of the F1 generation is what tells us which letter to use, what symbol to use for which allele. So in the case of Mendel's flowers, because the F1 generation was purple, and purple begins with the letter P, we are going to use a capital P to indicate purple and a lowercase p to indicate white. I often try, when the letters of the alphabet look the same in the capital and the lowercase, to make the lowercase cursive so that you can easily distinguish between the uppercase and the lowercase. So purple is the dominant allele, so it has the capital letter, and we choose P because purple is the dominant allele. Now, the hard, tricky part for beginning students is a lot of students want to change this to a lowercase w. You may not do that. You have to keep, we're still talking about the character, flower color, one character, one characteristic, 2 alleles, purple and white. Uppercase for the dominant allele, lowercase of the same letter for the recessive allele. So in this case, it would be capital P and lowercase p. So let's start at the beginning of this problem. Let's start with Mendel's true breeders. Okay? The P generation. And the P generation for this problem, remember, we had purple flowers crossed with white flowers that were true breeders. true breeders are going to be homozygous, so they're going to have two copies of the same allele. The purple flowers of the true breeders then would have two copies of capital P, and the true breeders for the white flowers would have two copies of lowercase p. Okay. But parents are 2N. They're going to produce gametes by meiosis, and that's going to separate these two alleles from each other in each parent. So I'm going to put one of the parents across the top, because this parent, the purple parent, can only put a capital P or the other capital P into, let's say, their pollen grains, and this parent can only put lowercase p or lowercase p into the ovules, into the eggs in the ovule. So I'm down the side, this is the other parent. The gametes for the other parent could possibly give to the offspring. When we fill in the Punnett square, basically what we're saying is what would happen if this pollen grain met up with this egg. What would our zygote, what would its genotype and phenotype be? Now there's one little rule about this. We always put the dominant allele first in the box, even if we're doing this. We're putting the capital P and the lowercase p. If this pollen grain meets up with this ovule, and we write it in the box like that, a capital P and a lowercase p. The reason we do that is because anytime we see the capital P, that's going to tell us the phenotype. In this case, the phenotype is going to be purple in the heterozygous. So for this box, we would again put capital P, lowercase p, capital P, lowercase p, capital P, lowercase p. Can you see that all of them are the same? They are all heterozygous. Now, these offspring here, that is the F1 generation. We would describe their phenotype as all purple. And if I asked you their genotype, remember that's the alleles they have, you would put heterozygous, capital P, lowercase p. Now, let's just continue this on to the second part of Mendel's experiments, where he allowed the F1 generation to self-fertilize to produce the F2 generation. Usually in the classroom, I would put on top of this drawing of the Punnett square the genotypes of the parents that I'm going to put inside this Punnett square. Today I'm just going to put it underneath. So for this Punnett square now, we are going to cross an F1 with an F1. So we're going to allow those F1s to, remember, we call that self-fertilize. Okay. The genotype, let's start with the phenotype. The phenotype of the F1s, remember, was all purple, but they were heterozygous. So the genotype of the F1s is going to be capital P, lowercase p, crossed with capital P, lowercase p, right? Because all the F1s were purple. Purple's the dominant allele. But the F1s are hybrids. They are heterozygotes. So they have one copy of the allele from each of their parents. Capital P from the purple parent and lowercase p from the white parent. Now, when we place the gametes on the top and the sides of our Punnett square, right, we're going to just look at this organism here. The possibilities that they could put into their sperm cells, say, their pollen grains, would be either a capital P or a lowercase p, right? Because they're heterozygous. They could put this one in or that one. And same for the egg or the ovule. We would have a capital P and a lowercase p. Then, just as before, we would fill in the zygotes, and those zygotes are going to be the F2 generation. So we would put, what if this pollen grain met up with this ovule? That would be capital P, capital P. If this pollen grain met up with this ovule, capital P, lowercase p. If these two got together, now again, remember that rule that I just told you, we're going to put the uppercase letter first, the dominant allele first, even though we would be tempted to put lowercase p first because it's right here close to us, right? So we're going to put capital P, lowercase p, and then in this last box, if this pollen grain met up with this ovule, I would have two lowercase p's. Next to the box, it is common practice to write out the phenotypes and count up how many of the F2 generation show each of the phenotypes. So I have the phenotype here, and one of the phenotypes is clearly purple. This offspring looks purple, so put one there. This offspring is heterozygous, but it's still purple, one there. And this offspring is also heterozygous, so that's 3. And then I have this F2 here, which has 2 copies of the lowercase p, so it's homozygous for the other allele, the recessive allele, which is going to be 1 white. So out of the total, we would say 3/4 of them are going to be purple and 1/4 are going to be white. I have 4 problems that I typed out for you as practice problems for a monohybrid cross. Again, we call this monohybrid cross because we're just looking at one character, one trait, in this case flower colored. I have used other P traits, other traits of the P's or characters of the P's in the problems and all of the vocabulary words. So I would advise you to watch the videos, read the chapter, and then be sure you print out the handout that has how to convert a word problem or a story problem in Mendelian genetics into the correct Punnett square to get the right answer.